How are we all doing today, everybody? Good so far? Okay, all right, I'll take it. I mean, the sun's been shining, so hopefully pretty good. Uh, all right, well, it is 3 o'clock, so we're going to go ahead and get started. I first and foremost want to thank you all for being here today at the Wild Center and supporting us and all the wonderful programming that we have to offer you all. And we have something special on the docket, which is why you're all in this room right now. Today is marking the second speaker for our Science Speaker Series of the summer. We are being graced by Jennifer Grauer, who's a PhD student at Cornell University, and they're going to be sharing about moose health and survival here in New York State, which if any of you have lived up here in the Adirondacks, you may know moose are a fairly elusive creature up here, and not so much maybe in some of our neighboring states. So we're going to learn a little more about that today. But while you're all here, I want to let you know that we do have other speakers joining us this summer. So if you're able to come back, we have Dr. James Peruk, who's going to be here on Thursday, July 20th, discussing his book, Loon Lessons, and his 3 decades of research into loons. And he will also be having a book signing afterwards. And then we have author Layla Phillip here. I always want to put an S at the end of that. Layla Phillip. He's going to be here on August 12th, showcasing her book, Beaverland, which is all about that one weird rodent that shaped America. And so we'll learn more about beavers and how they have helped to shape everything that we know and love in this country. So a couple of authors coming up as well as scientists. But without further ado, today is the day that we're talking about moose. So Jennifer here has been doing research for many years. She has researched everything from moose to martins to ants to lizards to deer and everything in between, and is now applying those skills to our Adirondack moose population. She has been with Cornell now for 2 years. Is this your second year? Yeah, and we'll be graduating with her PhD here shortly, just wrapping up her field research and then writing the paper. So we're very fortunate to have some of these preliminary research being shared here at the Wild Center. If you have questions, please keep those in your head. At the end, we'll have plenty of time to facilitate questions, and we would love to hear what you're wondering about as well. So without further ado, let me turn it over to the real star of the show today, Jennifer, to share with us a little bit more about her amazing research. All right, thank you.
Everybody hear me okay from this? Maybe this one? Yeah. All right, that's probably good. Okay, great. Well, thanks all so much for being here. I'm really excited to share everything that I know about moose in New York and then what we've been doing for our project and my PhD work for the last few years. And I am curious, so I'm going to ask for a little participation and show of hands. Who of you knew that we had moose in New York? A good group of you? Awesome. That's really good. That's more than I usually see. And have you ever seen a moose in New York? All right, so that's a good bit too. Yeah, definitely fewer than the people who knew, so not everybody, but that's a good number. And any guesses for how many moose we have? If you want to call out a number before I dive into some information. 500? Those are very good guesses. Yeah, you guys are all in the right range. And before I dive into what we know, I'll just start by introducing myself. Like Leanne said, I'm a PhD student at Cornell University, finishing up a lot of the data collection part of the project, and I work with advisors both in the Department of Natural Resources and Cornell's Vet College. And then the project involves a lot of partners in addition to that. So we work really closely with the NYSDEC, the Department of Conservation for the state, as well as U.S. Geological Survey, and they both provide different funding and resources for the project. There was a lot of research on moose before I even got to the state, so I'm kind of picking up where other projects have left off, where we're trying to get an idea of how the population is doing and what might be limiting them, kind of controlling the population growth in the state. And then also just a shout out to all the landowners who have helped me on the project. It's really been a mix of state and private land that I need to go to access some of the areas in the Adirondacks since it's such a mix. So we wouldn't have been able to do it without all these partners and people involved. But getting into the fun stuff. So those guesses were pretty spot on. You can see, let's see, down in this corner we have New England in the Northeast. And New York is they're right at the bottom, around 500 to 1,000. This was the estimate as of 2017. And what I also want to point out from this map, they're showing population sizes in different provinces and states. And the pluses and minuses are indicating whether the population is increasing, decreasing, or S for remaining stable. So you'll notice along the southern part of this, in the lower 48 states, the populations tend to be a lot smaller. There tend to be a lot more minuses, and so these tend to fluctuate a lot. And zooming in on the Northeast, we've had kind of a mixed history here for moose in the past, where historically they were distributed all the way down through Pennsylvania. Currently, the states occupied are these ones shown in gray, but they only really came back to these areas starting in the later 1900s. When the area was being colonized, there was a lot of forest conversion to agriculture and a lot of over-harvesting, which pretty much eliminated moose from everywhere but the northern portions of Maine. So now that they're back in these areas, and the current distribution is pretty much this dashed line here. So they're not all throughout these states, but these are the major areas where we're seeing them. Maine actually has some of the biggest populations outside of Canada and Alaska. So in the US, this is really a stronghold that we're seeing at their southern distribution. New Hampshire and Vermont also have pretty large populations, but they've been tending to fluctuate a lot, and the state agencies have kind of changed whether or not we can hunt versus not moose in that state. So here in New York, we have the DEC has been refining methods to estimate what the population looks like and keep track of this over time. So now we think there's maybe more in the realm of 700 to 800 moose, and we're refining that. But there's still a lot of questions as to whether the population is increasing, decreasing, or we think it's going to stabilize around this number. And so they really started recolonizing the Northeast after a spruce budworm outbreak in Maine that destroyed a lot of the trees and caused a lot of regeneration to occur. So moose had a lot of food to eat, and they started coming back into the Northeast, spreading into Vermont and New Hampshire, and finally New York after that. This was about the 1980s, where the DEC had kind of talked about assisted reintroductions into the state, but they didn't really largely need to do that at all. They kind of recolonized the area themselves. And so this is a figure just showing public sightings over time that has led us to believe that the population is really increasing and doing pretty well. But like I mentioned, they've been working with a couple different techniques to refine this and get a better estimate of the population. So this top one, the 716, that's from aerial surveys that the DEC will conduct every 3 to 5 years. And so they're flying in a plane, keeping track of the number of moose they see across transects and estimating distance from that transect to kind of qualify their estimate and get a better estimate of how many moose they think there are. This 549 comes from another survey where they collected moose pellets across the landscape and tried to estimate population that way. Then there have also been previous efforts to capture and collar the adult cows and then actually estimate how well they're surviving over time. So from this work through SUNY ESF, we were able to see that the adults are actually surviving really well. It's about 80 to 90% of those cows that are collared would survive to the next given year. They also kept track of pregnancy rates and then would go in and look at those cows that they figured out were pregnant to see if they were successful in reproducing and if they had just one calf or twins. So that actually looked really positive. We had high adult survival, high calf retention, and we know a couple other things about moose here that are adding to the story. So those collared moose, they actually went in and followed where they had been and saw where the moose were browsing, and then they estimated based on that what the amount of forage was available to them in New York, and found that while in the winter they have plenty of conifers to sustain themselves, in the summer they actually could be limited by the amounts of summer protein available. And then this could pose a challenge in the future for habitat management because of the forever wild clause that controls what can be managed with state land. there isn't necessarily additional land that can be used for, say, timber management, where you can cut down trees and create this regenerating habitat that mooses really respond to. We also know that climate change is happening. So there's a lot of different concerns for moose. And if we have, in general, increasing temperatures, heat stress can be a really important factor that can limit moose in the summers especially. So they have different behaviors that they can use to account for this, like spending time in wetlands, taking shelter in those conifer areas. But the future will see whether or not they can adapt to these different conditions that we'll likely see. So they certainly have a lot to contend with here. But there's still a lot of unanswered questions. So like I said, we're trying to find out and quantify actually, is the population increasing? Is it decreasing, or is it kind of stabilizing at this number that we're currently at that we think can be sustained based on that nutrition available? And we're also wondering if that summer protein alone is responsible for limiting the population to the numbers and low densities that we're currently seeing. So one thing that's influencing a lot of populations along that southern range limits are parasites, and we're wondering if moose are being affected by those here like they are in other states. And just in general, can New York sustain a healthy moose population into the future? And what are different management options and things that landowners can do to help? So zooming in on parasites, that's where my project is really focusing. And you've probably heard of winter ticks and the big effects that they can have on moose. And so the winter ticks seem really specialized on moose, where they'll infect deer and other animals, but they seem to do a good job of preening them and getting them off. Moose tend to have a harder time, and like this picture here shows, they experience a lot of hair loss from trying to groom off the ticks. And so the ticks will quest for a host in the fall, so they're getting up on vegetation, trying to be picked up by an animal. They'll infest the moose and then attach and stay there all winter. So the females will take a blood meal, and then in the spring, if they got enough blood, they will drop off and have their eggs, and the cycle will continue next fall. In the winter, moose can have like tens of thousands of these ticks on them in some of the really bad areas. And this can lead to a substantial amount of blood loss. And then when they're really low on hair like this, they can get a lot of thermal stress from winter conditions. So this is a really big issue for moose in Maine, especially where they have that high population with these really dense areas of moose. So it's kind of like a cycle where the ticks won't go very far after they drop off. And if it's a really moosey area, there's more and more moose there, and the cycle just propagates. So they're looking at a lot of different methods to try and combat this, and I'd be glad to talk about that more. But it is kind of hard to actually target the ticks themselves, so they're potentially looking at reducing the moose density to see if that works. But there's also a lot of other parasites that moose have to contend with. So these are all internal parasites that I'll be talking about, where we have a giant liver fluke that pretty much does what it sounds like. It hangs out on the livers, it makes these cysts, and it can cause a lot of damage. Both this and the brainworm actually go through white-tailed deer normally as their hosts, but they can cause a lot of damage in these incidental hosts like moose and other ungulates. The brainworm in particular, you might have also heard of. It's a worm that kind of migrates to the brains, and in moose it can cause a lot of neurological issues, and both of these can ultimately cause a lot of organ damage and sometimes death. They also have a lot of other fun things to deal with. So they have tapeworms, and this one species, which we don't have a common name for, is just called Neospora caninum. These both go through canids and then usually ungulates as in a multi-host life cycle. And lungworm, so all of these three can also cause a lot of inflammation. They can cause damage to different organs. And then some of them have their own little special diseases, like Neospora can cause fetal abortion in some of the moose. So a lot more to deal with. And the ones I'm focusing on more for my project are primarily the tick, the brainworm, and the liver fluke. these seem to be a lot more prominent and prevalent and causing issues and declines in moose population elsewhere, especially Maine, Minnesota. So I wanted to mention a little more about the life cycle for these parasites, and this one is brainworm. So wherever you see this little guy, I'm speaking to brainworm. And this is a nice figure that the DEC put together about their life cycle where, like I mentioned, the deer are their primary hosts, and then parasites are shed from their feces. They'll get taken up by slugs and snails across the ground, and then they develop within the slugs and snails to an infective stage, which then goes back up into deer, and the cycle continues. When moose pick these up, this is when the moose can get infected, have neurological issues. You might see them stumbling around, sometimes circling a lot, and they're not really able to take care of themselves at that point. So they might get hit by a vehicle, they might have to be euthanized by a DEC officer, or they just end up starving. This brainworm, the similar kind of figure and similar life cycle, but these ones actually go through aquatic snails and slugs. So feces will go into water bodies, they'll make their way into a snail, and then they grow inside the snail. They get shed from that snail, and there's a free living stage, and that gets taken up again by the deer or other animals. And then this figure here is showing an infected versus a normal looking liver, where this dark red one on the right is the healthy one, and then the pale one with these kind of dark lesions and things on them is an unhealthy infected one. So I probably don't have to mention this to you all because you're here and you're very interested, but just to kind of go over why we really want to do something about this and why we want to investigate what's going on. So first and foremost, it is the DEC's job, their role as a state agency, to preserve wildlife and natural resources for all citizens and also future generations. So this is them trying to learn more about the population and do research to see how we can sustain them. They're also really important ecologically. So because of how much they eat and how much they move nutrients around on the landscape, they're really functioning as shapers of forest ecosystems. So a lot of this exchange determines what other animals and plants are in an area and how forests regenerate. Then also being in the Adirondacks, I'm sure you've seen that they're very culturally important. They're definitely a big icon, important to the North Woods feel, and then they're really big for tourism. So specifically with my project, we're trying to get at this rate of juvenile survival. Since we know pretty well how the adults are doing, we'd like to know a little bit better if the juveniles are surviving well and if this could possibly be limiting the population. We also want to determine the impact of those parasites on both individuals and at the population level. And then understand how moose during their day-to-day are kind of dealing with this infection risk. We're also using a lot of different methods to study them and seeing which ones the DEC can use in the future for their monitoring, and trying to analyze which of the multiple management options available to them could actually be helpful in reaching their goals for moose conservation. So this border here is the outline of the Adirondack Park, and what you can see, these black dots are moose observations and the red dots are deer observations from those aerial surveys. And so we wanted to focus in this gridded area, and the northern portion is where I've kind of focused my survey efforts. And a lot of that is because this is where we have the highest density of moose in the state, and we also see overlap of moose and deer. And so within this gridded area, this is just that zoomed in. We're looking at 3 different areas and doing our sampling across each of these little grid cells. So we have different kinds of habitat available, different densities of both moose and deer. One of the methods we're using is putting out trail cameras. So we have 105 of those grid cells. We're putting out 1 trail camera in each of those. And we've actually had these out for about 2 years now and are pulling them this summer. And so what this can help us look at is both estimates for the moose abundance and deer abundance across the study area. And we're also trying to see if we can use the cameras to monitor for the effects of the winter ticks, if we can actually see hair loss, usually in the late winter, early spring, on any of the moose. We're also doing capture and collaring efforts as part of this juvenile survival study. So both this past January and the January of 2022, we worked with a capture team, which goes out in a helicopter and will actually use a net gun or dart gun to sedate and then put collars on these moose. So we're targeting ones that are calves from the previous summer, and those are about 6 to 8 months old at this point in the winter. And then the idea is to monitor those for the next 2.5 years or so, which is how long the battery on those collars will last, and then keep track of how many are surviving, and then if they're not surviving, what is the actual cause of death. We're also walking around conducting a lot of those pellet transects, but this time for white-tailed deer pellets. And this is because with those two internal parasites, the liver fluke and the brainworm, the parasite eggs and larvae can't actually pass through the moose and complete their life cycle, so they don't get shed in moose pellets, but they do get shed in deer pellets. So we've collected a lot of those, and that can tell us what parasite prevalence across the landscape looks like, kind of as a way to look at infection risk for moose. And finally, we're doing another kind of test of methods using environmental DNA. And so environmental DNA is basically the DNA that gets shed from you, from any animals, and is left in the environment. So we could take, say, a piece of sticky paper across the stage and then see who was here, what kind of animals were here. And we can do the same thing in water bodies. So we actually went out and collected water, and then we would filter it onto these little filter papers, which I can then do genetic methods to extract the DNA from there and tell what was in that certain area. And with this in particular, I'm looking for the DNA of those liver flukes. So we're capturing, hopefully, that eDNA when they're either the larva stage, free swimming around, or the actual eggs themselves when they're a little earlier in age. And this also informs, infection risk and what kind of prevalences we see of liver flukes on the landscape that moose have to deal with. So right now I'm kind of wrapping up all the data collection for the project. So I don't have answers to a lot of these questions yet, but what I can share with you is some of the preliminary results that I've been finding and what I've done so far. And what have we seen? So there's pretty much deer everywhere. This wasn't always the case. I've definitely heard from folks that, growing up, they didn't used to see as many deer. The DEC would tell me stories about how they migrate seasonally in and out of these high snow areas, but now they pretty much seem to be ubiquitous throughout the Adirondacks. Might surprise some hunters, but they're around. We're also seeing a lot of bears. This was really cool. I think they're just so fun because we saw lots of cubs wandering around, and they really enjoyed using these snow stakes for scratch posts. We're also seeing pretty much any mid-sized creature that was around the Adirondacks. So we got to see turkeys. Does anyone know what this on the right is? Yeah, the fisher? Yep. We've seen bobcats, coyotes, foxes, everything except the really small things because we turned down the sensitivity on these cameras, so we were just capturing the big stuff. But we did see a lot of moose, so this was really exciting too. Sometimes they would just be passing through like this, and we got to see a lot of moms with their calves. They were looking, on the most part, pretty healthy. Sometimes they would hunker down, and we got to see really cool photos like this too of them nursing right in front of the camera set. And then sometimes they would just hunker down for a nap. But by and large, we were seeing kind of what we expected from that first figure of the study area and where we saw a lot of observations. They're pretty much limited to these areas of prime habitat where we have regenerating forest stands. We also are seeing some mild cases of the hair loss. So this is just one that one of the landowners sent me of moose in their front yard, but right around that late spring time of year when we would see that hair loss. We've seen maybe another dozen or so from the cameras that are showing this as well. With the survival study, between those 2 Januaries, we were able to collar 31 individuals, and they all looked like they were in really good body condition. And this is a positive thing. You know, there's enough food to support them. They're doing pretty well, but they did all have winter ticks on them. So we would actually count the ticks that we saw on transects on the moose's shoulder and on the rump. And this is different from what we saw during that first collaring effort with the adults, where maybe 1 or 2 had ticks on them, but these last few years, all of the moose had had ticks on them. We've also seen 6 mortalities so far. We had 1 last summer, and then this spring and early summer, we've had 5 already. I do want to talk a little bit more about these mortalities. So if you're squeamish, this is just a warning that there are some dead moose photos on the next few slides. And so what we're seeing, when a necropsy or when a moose dies that's collared, we'll go and try and collect the whole carcass. Then we'll bring it to the DEC lab for them to do what's called a necropsy. And so that's basically an autopsy like would be done on a human to determine cause of death. And what we're seeing so far is that they're all infected with these liver flukes, and they have varying degrees of damage to the liver from these. So here, the liver looks kind of okay, like coloration is decent. All of these little dark things that look like pieces of liver are actually those liver flukes. So these guys would be just all infested in the livers, and then they change the consistency to almost like a chocolate pudding. So it's not great. When we can't collect the whole moose and bring it in, we end up doing what's called a field necropsy. So just using what we have, kind of bring everything out, and then doing this in the field. This was a moose that died last summer, and she did have some evidence of the winter tick hair loss on her. And then what you can see, what you're looking at here is a lot of the gastrointestinal tract for a moose. But what I draw your attention to is this, is actually the moose's liver. So it's not recognizably what a liver should look like. And this had a really bad infestation, damage to the liver. And actually all of these black trails that you're seeing are evidence of the flukes migrating around the body. So they cause a lot of damage. They can cause liver failure and death. And we're seeing that a lot. That's it for the gruesome photos. I'll move on. But we've also been able to look at data from the past 20 or so years when the DEC conducts necropsies for moose that they find dead. And these are usually either vehicle collisions or sometimes when a moose is irreparably injured and it needs to be euthanized. So this can actually be helpful to look back on and collect information on the health of moose, looking again at those causes of death, and then getting information on parasite prevalence. And so this is a map of all the necropsies that were done between 2000 and 2022. So we had almost 200 of those cases that we can pull information from. And the colors just show the different numbers that were in each of these different townships. The black dots here are also the locations of all of those collared moose. So just to give you an idea of kind of where they're occurring. What we saw from this, this is showing year across the bottom axis and the proportion of necropsies in a given year on the y-axis. And what we're seeing, all of these tan bars are sources of trauma. So when a moose dies from a vehicle collision or from either poaching, sometimes they do get shot in poaching or self-defense, or if they're euthanized by the DEC. But the second most prominent category was parasites. These are that light gray bar. And so these were cases where they could explicitly say it was either liver fluke or brainworm that was the source of death. We also have information on the numbers of parasite, or the cases that we were seeing. So by and far, 40%, we're seeing the most cases of liver fluke. Brainworm, definitely less so, but we are still seeing high rates of exposure from some of the blood work that we've done. The other parasites, definitely less so, but they're still around. So with all of this data, I wanted to see, based on the locations of those necropsies, what kind of factors and things going on in the area were really important to determine if a moose was infected with the liver fluke or the brainworm. So we looked at different things like road density in the area, white-tailed deer density, percents of deciduous forests and conifer forests and wetlands. And when we did this for brainworm, the biggest thing that was most important for whether a moose was infected with brainworm was white-tailed deer density in the area. And this seems pretty intuitive, right? Because they need to go through the white-tailed deer to even be in the area. And this is predictions. The error bars aren't showing up, but these are predictions at different, the range of white-tailed deer densities that we're seeing across the Adirondacks. So as you can see, there's a positive relationship. When there's more deer in the area, there's more risk for moose. When we looked at this for liver fluke, deer didn't really come up at all, which was kind of interesting. Instead, we were seeing differences based on how old a moose was. So in the red, you have adults with just in general a higher chance of being infected, and the blue is the juveniles. So this might happen just because adults have more time to get infected with parasites. They might be covering more ground, all of those things. And then it was also interesting that we saw a trend of increasing risk over time. So 0 here is the year 2000, and it goes up to 2022, where we're seeing with time, moose are just having higher chances of being infected. And the big things we take from this is that obviously different things are important for infection of those different species, but this is really good evidence that deer are really important and definitely need to be taken into consideration. Also, it's kind of alarming that this is an increasing trend over time, so definitely something that should be monitored. And in general, there's different ways with parasites to kind of manage these things. So different things that you can manage would be, one, the parasites themselves, whether it be treating for winter ticks, trying to somehow across the Adirondacks decrease the numbers of liver flukes and brainworm, which sounds just, about as hard as it could be. We could also then talk about managing the hosts. So this would either be managing the moose themselves or managing white-tailed deer. And then the other option is to manage the environment. So kind of controlling which areas are usually going to be more likely to have the brainworm in them or the liver fluke. This is also pretty difficult since, one, the Adirondacks are so large, but it does seem that the most feasible option, especially right now for the DEC, would be to manage for white-tailed deer. And so if we can separate the areas that the moose and deer are really overlapping in and understand that more, that might be the most tangible way we have for addressing this currently. But there's a lot more that needs to go into this. I'm certainly going to analyze this and try to come out with more tangible options for the DEC. Some of that's going to be doing population analyses once we know more how well the juveniles are surviving over the next few years. And also actually seeing if they are dying from parasites like we think they are, how much of an impact does that have at the population level? Also, once we have that information and I can do different simulations to see what certain DEC management options could have for potential outcomes, we can compare these different management options. So that's what I'm currently working on. And all of this, like I said, part of this is funded by the DEC, so they really are interested in using this information to help craft their moose management plan for the state. and help with conservation of moose for the future. And with that, I'd definitely like to thank all the DEC folks that have helped with this project, as well as everyone at Cornell, my advisors and lab mates, and all of the technicians and volunteers that have helped with the project so far. So with that, I'd be glad to take any questions and chat further. Yeah. So where I would recommend, and the one place that I've seen them so far, is north of Saranac Lake, about an hour. And if you're familiar with, it's called like the Chateauguay Woodlands, is what the area is called, where there's a lot of active timber harvests. There's dirt roads, there's wetlands, kind of everything that you would need to access the area and see these really moosey habitat types. That's where I would recommend.